The plastics industry is huge. Over 335 million tons of plastic are produced globally each year, and that number's only getting bigger. However, a large majority of plastics are derived from petrochemicals, which are a finite resource. The day we run out of crude oil is one that many of us will live to see, and as a result there is interest in developing ways to derive polymer feedstocks from renewable resources. To understand where we should begin looking for renewable feedstocks, we'll need an understanding of what polymers are made from. Polymers are long chains made up of many smaller molecules called monomers. They're often categorized by the type of functional group that links the monomers together. Three important types of polymer are polyalkenes, polyesters, and polyamides. Polyalkenes are, as the name suggests, made from alkenes. In the polymerization process, carbon-carbon pi bonds are broken to form sigma bonds with other monomers in a chain reaction. Because of this, alkenes are the only monomer necessary. Polyesters are polymers where the monomers are joined together by ester bonds, which are made from carboxylic acids and alcohols. In order to form a long chain, the monomers must be either a mixture of dicarboxylic acids and diols, or a single polyfunctional monomer that contains both groups. Polyamides are quite similar to polyesters, but monomers are joined together by amide bonds, which are made from carboxylic acids and amines. Therefore, long chains must be made from either a mixture of dicarboxylic acids and diamines, or amino acids. Now that we know how polymers are made, we can start looking for renewable monomers. One promising place to look is in the metabolic products of living organisms. Almost all organisms get their energy from the oxidation of glucose, and since glucose contains 5 alcohol groups, it's reasonable to assume that metabolites of glucose will also contain alcohol groups, or acid groups, since acid groups can be thought of as the oxidized form of primary alcohols. One such metabolite is lactic acid. Lactic acid is synthesized by humans during anaerobic respiration. It's not exactly feasible to harvest lactic acid from humans, so on an industrial scale it's derived from the fermentation of bacteria. As it contains an alcohol group and a carboxylic acid group, it can be polymerized into a polyester. Polylactic acid has highly tunable properties due to its chiral nature. A 1:1 mixture of poly-L-lactic acid and poly-D-lactic acid has a melting temperature up to 50 degrees Celsius higher than pure poly-L-lactic acid. It's biodegradable and has potential applications in medicine as temporary implants because the human body is capable of metabolizing polylactic acid on its own. Another approach is to chemically modify renewable chemicals that are not polymerizable on their own into ones that are. One such molecule is ethanol, which humans have been making by fermenting yeast for millennia. Ethanol itself only contains one alcohol group, so it cannot function as a monomer. But, under acidic conditions, a water molecule can be eliminated from ethanol, giving ethene. As an alkene, this can be polymerized into polyethylene, which is the most common plastic, though most of it's derived from petrochemicals nowadays. Polyethylene also has highly tunable properties that arise from modifications made during the polymerization step, such as introducing branched chains or crosslinks. We can also get more creative with our chemical modifications. Castor oil, which is a plant-derived oil's main constituent is a type of fat called a triglyceride, which consists of 3 fatty acids ester bonded to a glycerol molecule. In this particular instance, hydrolyzing the ester bonds gives 3 molecules of ricinoleic acid, and after a long, complicated reaction process involving an esterification with methanol followed by thermal cracking, another ester hydrolysis, an anti-Markov-Nicovian addition of hydrogen bromide in the presence of a peroxide, and a nucleophilic substitution with ammonia finally gives 11-amino-undecanoic acid. Being an amino acid, this can be polymerized on its own into the polyamide nylon 11, which has several high-performance applications, such as pipes for high-pressure fluids and gears. These so-called bioplastics have the obvious advantage of coming from renewable feedstocks, and being mainly plant-derived, they have a significantly smaller carbon footprint than petrochemical-derived plastics. This is because the sugars and carbohydrates used as raw material are made from carbon dioxide by plants during photosynthesis. However, it's not all good news. The plant derivatives used in many of the fermentation processes used to make the monomers are derived from edible crops, such as sugarcane, corn, or wheat. This means the feedstocks are in direct competition with food, which may lead to sustainability issues further down the line. Even in cases where the primary resources are not edible, such as castor oil, there is risk of deforestation and competition for land that would otherwise be used to grow food. In the future, interest in bioplastics is sure to increase as petrochemicals become more and more scarce. But what direction will bioplastics research go from here? One promising area of interest lies within the field of genetic engineering. For example, we can alter the genes for specific desired enzymes to control the fermentation product. In the production of lactic acid, the bacteria naturally produce a racemic mixture of lactic acids. However, since the enzymes that make lactic acid are stereospecific, removing the gene for one particular type of lactate dehydrogenase enzyme results in an antimerically pure D or L lactic acid. Another area that is currently being researched is engineering microorganisms that can synthesize metabolites that are not naturally produced. For example, the polyester polybutylene succinate is made from 2 monomers, succinic acid and 1,4-butanediol. Succinic acid can be made in bacterial fermentation, but 1,4-butanediol is essentially a highly reduced form of succinic acid, and is very unlikely to occur naturally due to the oxidizing nature of cellular respiration. Indeed, it's not made by any known naturally occurring organism, and 1,4-butanediol is currently derived from petrochemicals. However, by using computer software to simulate possible metabolic pathways to 1,4-butanediol, researchers have been able to engineer a strain of E. coli bacteria that can synthesize 1,4-butanediol from glucose.